Modeling meiosis. A diploid cell will duplicate in interphase before entering meiosis. Meiosis occurs in two parts, 1 and 2. Meiosis 1 is the reduction division where the diploid cell is divided into two haploid cells, each containing 23 chromosomes, unlike mitosis where the number of chromosomes remains the same. The first step of meiosis 1 is prophase 1, where homologous chromosomes pair up. Homologous chromosomes have the same genes, but they do not necessarily have the same DNA sequence and are not connected. In prophase 1, the most important event occurs here, crossing over. Homologous chromosomes pair up to form a tetrad. The chromosome pair crosses over at the chiasma, which means that they exchange genetic material between each other.
The second step of meiosis 1 is metaphase 1, where recombined homologous chromosomes line up in the center due to spindle fibers. Then, in anaphase 1, the recombined homologous chromosomes relocate to opposite sides of the cell, resulting in shorter spindle fibers. Finally, telophase 1 and cytokinesis occur. A cleavage pair forms, dividing the cell into two separate haploid cells. There is one copy of each chromosome and is duplicated with two chromatids.
Meiosis 2 follows shortly after meiosis 1. Between parts 1 and 2 of meiosis, there is no interphase because the DNA is already replicated, but there is a brief pause. Meiosis 2 is very similar to mitosis because the process is identical. However, meiosis 2 forms 4 distinct haploid cells, whereas mitosis results in 2 identical diploid cells. In prophase 2, the cell prepares to break down once again. The nuclear envelope containing the chromosomes begins to fade and dissolve. During metaphase 2, the chromosomes line up in the middle yet again because of the spindle fibers. Then in anaphase 2, the spindle fibers pull the chromosomes apart of their centromeres, creating chromatids at opposite ends of the cell. Finally, telophase 2 and cytokinesis occur, forming 4 cells with one sister chromatid of each chromosome in them.
Errors in meiosis can often arise, such as non-disjunctions. A non-disjunction is when chromosomes or sister chromatids fail to separate and is usually diagnosed via karyotype. One instance is non-disjunction appearing in meiosis 2. The cell goes through meiosis 1 efficiently, but when it reaches anaphase 2 and meiosis 2, non-disjunction is apparent. One chromosome does not separate while the others do, causing one haploid cell to have an extra sister chromatid. Non-disjunction of meiosis 2 affects half of the gametes. If the gamete is fertilized, various disorders such as Turner syndrome, Jacobs syndrome, and trisomy X can develop. Genetic diversity is an exclusive characteristic of meiosis. Crossing over is one way that meiosis contributes to genetic diversity, which is the differences in genomes amongst a population. New combinations of genes are produced, which makes each species unique. Crossing over is not an option in mitosis because mitosis creates a complete clone of itself.
Independent assortment also provides genetic variety. It describes the combination of maternal and paternal chromosomes that make up the chromosomes set in the haploid gamete. In our model, 8 different combos can be produced by the independent assortment alone because there are 3 haploid cells and 2 to the third in this case is equal to 8 possible combinations.